Hello. Now we'll begin Chapter 7, which focuses on nerve and synapse physiology. So we'll begin with looking at the brain and looking at the neurons and supporting cells. So if we look at the nervous system, we can divide it into the central nervous system, which consists of the brain and spinal cord, and the peripheral nervous system, which is composed of the cranial and spinal nerves. Tissues in the nervous system are notably classified in two types. One type are neurons. The neurons are cells that conduct impulses, and these cells can generally not divide. And there are also glial cells, also called neuroglia, that support neurons and cannot conduct impulses, but these cell types can divide. So if we look at this image of the neuron, I want to point out that it has a cell body, and inside the cell body we see a nucleus. There are dendrites that can basically receive information that we'll be talking about later in this presentation, and the processing of either inhibitory or excitatory signals will basically add up to giving either a positive or excitatory or inhibitory signal at this portion of the cell body called the axon hillock. And the axon hillock is where the nerve will make, or neuron will make a decision. Will it create an action potential? And then here, going away from the cell body, we see an axon, and this happens to be a myelinated axon, and there are these myelinated regions, which are basically insulated regions that allow an action potential to jump from the axon hillock to the node of Ranvier, and to one node, another node, etc., until the end of the axon is reached. So what are neurons? So neurons are specialized cells in the nervous system. They can respond to chemical or physical stimuli. They can conduct electrochemical impulses. They can release chemical regulators, which we call neurotransmitters. And they play a critical role in perception, learning, all the senses. They play a role in memory and control muscles and glands. And again, neurons and nerves, most of them cannot divide. However, they can be repaired in the peripheral nervous system with the help of some glial cells. And I want to point out that axons can be either short or they can be very long. So a short axon would be a few millimeters. Some of the longer axons in the body are actually over 1 meter in length. And in the case of a really long axon, this myelination plays a really important role because it allows the nerve impulse to travel over a long distance, for example, over a meter, to send an impulse over that distance very quickly. So if we look at the classifications of neurons, we can classify them as sensory neurons, and sensory neurons can conduct impulses from sensory receptors, and they bring that information to the central nervous system. Motor neurons conduct impulses in the opposite direction. They go from the central nervous system to target organs such as muscles or glands. And then there are also interneurons, and interneurons are located completely within the central nervous system, and they serve an integrative role to help integrate different signals coming to and from the central nervous system. So I mentioned that there are glial cells, and glial cells are the neuroglia, and these are non-conducting support cells that help support neurons. And in the peripheral nervous system, we have Schwann cells, and Schwann cells have the ability to form a myelin sheath, which I showed you in the picture around the peripheral nervous system axons. And there are also satellite cells that help support cell bodies within ganglia. Ganglia are like a gathering or having many nerves in one area of the peripheral nervous system. So if we look at the glia in the central nervous system, they are actually quite distinct and different than the glia found in the peripheral nervous system. So let's take a closer look at some of these. So in the peripheral nervous system, I mentioned to you that the Schwann cells have the ability to myelinate the axons. And in contrast, in the central nervous system, we have oligodendrocytes that perform this function. So for oligodendrocyte, there can be one oligodendrocyte. It can actually have these multiple extensions that can wrap myelin around different axons. So here's one portion, here's another portion of the same axon. But you can also see over here, there's a different axon that has the myelination. So these oligodendrocytes are really important, and they can put myelin sheath on multiple bundles in one axon, or even reach to multiple axons in the vicinity. Another specialized cell type found in the central nervous system are the microglia, and the microglia are part of the immune system in the brain. So the brain has an attenuated immune system because if the immune system was to function fully in the central nervous system, inflammatory responses could cause a lot of damage. So instead of having the full immune system functioning in the brain, they have some other factors like microglia who play an immune role, but they do not trigger massive inflammation. So we actually call the brain an immune privileged site because it doesn't have the full functioning of the immune system. However, it does have immunity, and the microglia can phagocytose, which means they can eat or destroy foreign material or maybe a pathogen that got into the central nervous system. And they are very important for a healthy brain. The next cell I want to talk about are these ependymal cells. The ependymal cells line the ventricles in the brain and central nervous system, and they secrete cerebrospinal fluid. So here you can see the cerebrospinal fluid, and that is being secreted by these ependymal cells. Astrocytes are another specialized cell that are found in the central nervous system, and these play a really important role in regulating the external environment. And these are also the most abundant glial cells. So these have these processes with these end feet that can reach onto both capillaries that are going through the central nervous system and also onto neurons. And what these astrocytes do is they actually facilitate interactions between neurons and the blood. So one important thing the astrocytes do is they take up potassium from the extracellular environment, and they help to maintain a proper ionic environment for the neuron. So they maintain an isotonic state in the central nervous system. And this is going to be very important because as we learn about action potentials, there's going to be huge changes in in ions moving in and out of cells in the central nervous system. The astrocytes, notably, can also take up excess neurotransmitter and basically pick them up and then recycle them. In terms of interacting with the capillary, what they do is they actually uptake glucose from capillaries, they convert it to lactic acid, and they provide the lactic acid to the neurons, and the neurons will then basically metabolize the lactic acid and produce ATP. Astrocytes can also store glycogen, and then let's say, if an astrocyte has excess glucose, they can store the excess glucose in the form of glycogen, and then when glucose levels go low, they can then break down that glycogen, reconvert it back to glucose, and then produce lactic acid. So astrocytes are really important because they're absolutely critical for the formation of synapses in the central nervous system. They regulate the processes of neurogenesis, which is nerve growth and nerve proliferation in developing humans and animals. They also help to regulate the blood-brain barrier, which we'll talk about in a few minutes. And they also secrete these special transmitter molecules that can stimulate or inhibit neurons. And again, during development of the central nervous system, they really help neurons migrate to the right location in the brain. So let's look at the blood-brain barrier. So the blood-brain barrier is really important because this protects the brain from having molecules escape the capillaries with little spaces between epithelial cells like it can normally do throughout the rest of the body, and it can't do this because the brain is a protected area, because since the brain doesn't have full immunity with all the inflammatory processes that come with that, the brain needs to be protected from having microbes enter the brain. So the blood-brain barrier happens because the epithelial cells lining capillaries have tight junctions between them. So the only way for substances to leave capillaries and get into the central nervous system is to have transcellular transport. Transcellular means that the molecules are being directly passing through the epithelial cell lining the capillary and crossing it to get to the other side into the central nervous system. So for example, glucose will leave the capillary, pass the plasma membranes of the epithelial cells, and then pass into the astrocyte. And paracellular is a term for going between cells, but because there are tight junctions between the epithelial cells lining the capillary, molecules cannot pass between cells, which is called paracellular transport. They can only move transcellular transport, which is going directly across the cells lining the capillary. And I should also mention that the blood-brain barrier sometimes is a problem for chemotherapy, because sometimes there are some drugs that do not have the ability to pass the blood-brain barrier.